Welcome back, my dear students. Today we are going to deliver the fascinating world of pentose phosphate pathway, which is also known as the hexose monophosphate shunt. It's a vital metabolic route that runs parallel to the glycolysis. This pathway is not just about breaking down glucose, but about producing NADPH, the cell's reducing power, and ribose 5-phosphate, the key building block for nucleic acids. Through its oxidative and non-oxidative phases, the pentose phosphate pathway supports biosynthetic reactions, antioxidant defense, and cellular growth. By the end of this lecture or class, you will clearly understand how the pentose phosphate pathway contributes to energy balance, cell protection, and metabolism, providing that glucose has much more to offer than just energy. So, the pentose phosphate pathway is an alternative way for the body to break down glucose and to separate from glycolysis and the TCA or Krebs cycle. Basically, it occurs in cytoplasm of both prokaryotic and eukaryotic cells. This pathway is important because it helps to produce NADPH, which is basically a powerful reducing agent, and ribose 5-phosphate. It's a sugar which is needed to make DNA and RNA. So the pentose phosphate pathway is also known as hexose monophosphate shunt. It occurs in virtually all living cell types and tissues. In liver, 30% glucose metabolized by the phosphate pentose phosphate pathway, and the process occurs in the cytoplasm. This process produces NADPH, which is necessary for several pathways like fatty acid synthesis for 50%, oxidative stress homeostasis, and cytochrome P450 enzymes. This pathway produces trioses, hexoses, and pentoses, and the pentoses are necessary for nucleotide biosynthesis. Now, we can discuss the phases of the pentose phosphate pathway. Basically, the pentose phosphate pathway has two main phases: oxidative phase and non-oxidative phase. Oxidative phase or irreversible reactions. This phase oxidizes glucose and generates NADPH. Let's go step by step. Glucose is converted into glucose 6-phosphate. This is the enzyme hexokinase. What happens? One ATP is used to convert glucose into glucose 6-phosphate, which is same as in glycolysis. The glucose 6-phosphate is then converted into 6-phosphogluconolactone. The enzyme needed for this reaction is glucose 6-phosphate dehydrogenase or G6PD. What happens? Glucose 6-phosphate is oxidized to form 6-phosphogluconolactone and 1 NADPH is produced. Then 6-phosphogluconolactone will be produced into 6-phosphogluconate with the help of the enzyme hydrolase. What happens? Hydrolysis of 6-phosphogluconolactone converts 6-phosphogluconolactone into 6-phosphogluconate, and the 6-phosphogluconate with the help of the enzyme 6-phosphogluconate dehydrogenase, it produces ribulose 5-phosphate. The compound is oxidized and decarboxylated, forming ribulose 5-phosphate and another NADPH, and the end products of this phase is the 2 NADPH molecule and 1 ribulose 5-phosphate molecule. This is the reaction steps of the oxidative phase of the pentose phosphate pathway. The first of all, the glucose 6-phosphate with the help of the enzyme glucose 6-phosphate dehydrogenase, the NADP plus will be converted into NADPH plus H plus, and it will produce 6-phosphogluconolactone. Then with the help of the enzyme lactonase, it reacts with the water molecule, and with the help of magnesium 2 plus ion, it will produce 6-phosphogluconate. Then the 6-phosphogluconate dehydrogenase again reduce the substance and produce the ribulose 5-phosphate with the help of the enzyme 6-phosphogluconate dehydrogenase, and in this reaction one molecule of carbon dioxide is released and NADP plus is converted into NADPH plus H plus. Then the ribulose 5-phosphate with the help of the enzyme phosphopentose isomerase, it will form ribose 5-phosphate, and the ribphosphate another epimer of the ribose 5-phosphate is xylulose 5-phosphate. So with the help of the enzyme epimerase, ribose 5-phosphate will be converted into xylulose 5-phosphate. And in non-oxidative phase, which is the reversible reactions, we can find that from the ribose with the help of the enzyme isomerase and epimerase, ribose 5-phosphate will be converted into xylulose 5-phosphate, and with the help of the enzyme transketolase, the ribose 5-phosphate will produce pseudoheptulose 7-phosphate, and xylulose 5-phosphate will produce glyceraldehyde 3-phosphate. Then again with the help of the enzyme transaldolase, pseudoheptulose 7-phosphate will produce fructose 6-phosphate, and glyceraldehyde 3-phosphate will produce erythrose 4-phosphate. Then fructose 6-phosphate with the enzyme phosphohexose isomerase, it will produce glucose 6-phosphate, and the erythrose 4-phosphate with the help of the enzyme transketolase produce fructose 6-phosphate, and fructose 6-phosphate will be converted into glucose 6-phosphate with the enzyme phosphohexoisomerase. And the xylulose 5-phosphate again with the help of transketolase will produce glyceraldehyde 6-phosphate will again converted into fructose 6-phosphate. So in the 4 compounds of the non-oxidative phases like fructose 6-phosphate, glucose 6-phosphate, fructose 6-phosphate another, and the glyceraldehyde 6-phosphate, they will enter into the glycolysis process to produce ATP. So what happens in this phase? The non-oxidative phase, which is also known as the reversible phase, it rearranges sugar molecules to produce a variety of carbon skeletons useful for other metabolic pathways, such as ribulose 5-phosphate with the help of the enzyme phosphopentose epimerase, it will convert into xylulose 5-phosphate or ribose 5-phosphate. Then transfer reactions, the reactions shuffle carbon atoms between the sugar molecule xylulose 5-phosphate plus ribose 5-phosphate. They will form pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate with the help of the enzyme transketolase. Again, pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate will produce fructose 6-phosphate and erythrose 4-phosphate with the help of the enzyme transaldolase. And xylulose 5-phosphate and erythrose 4-phosphate will produce with the help of the enzyme transketolase, they will form fructose 6-phosphate and glyceraldehyde 3-phosphate. So end products of these reactions are fructose 6-phosphate, glyceraldehyde 3-phosphate. Both can enter glycolysis or gluconeogenesis. Now we should know what pentose phosphate pathway, which is also known as hexose monophosphate shunt, generates. It mainly generates 3 types of molecules. Ribose 5-phosphate is necessary for the production of nucleotides, which ultimately produce the nucleic acid. Then NADPH, which is a driver of anabolic reactions like reductive biosynthesis pathway such as fatty acid synthesis. NADPH is also act as reducing agent. Both the NADPH also perform the role in oxidative stress homeostasis. And the third molecule is erythrose 4-phosphate, which is produced from the pentose phosphate pathway, and it is needed for the synthesis of aromatic amino acids. So now we need to know what is the significance of the pentose phosphate pathway. Main source of NADPH. The pentose phosphate pathway is the only cytoplasmic pathway that produces NADPH used in the biosynthetic or anabolic reactions. NADPH as reducing agent. NADPH helps in fatty acid synthesis, cholesterol synthesis, and detoxification of reactive oxygen species. Formation of ribose 5-phosphate, it is used to make nucleotides, the building blocks of DNA and RNA. It works in cells without mitochondria. Cells like RBCs depend completely on the pentose phosphate pathway for NADPH production since they lack mitochondria. It produces various sugars. It generates 3-carbon to 7-carbon sugars like triose, tetrose, pentose, and hexose and heptose for different metabolic needs. Role in detoxification. What NADPH does, it helps regenerate glutathione, which neutralizes hydrogen peroxide H2O2 and protects cell from the oxidative damage. Role in malaria resistance. People with G6PD deficiency, glyceraldehyde 6-phosphate dehydrogenase deficiency, which is common in some African populations, they produce less NADPH. So as a result, malaria parasite cannot survive well inside the RBCs, giving natural protection against malaria. So we need to know why pentose phosphate pathway is also called as hexose monophosphate shunt, because in these reaction steps we know from the glucose 6-phosphate is produced, and then further fructose 6-phosphate will produce, and the pyruvate. And this during pentose phosphate pathway, with the help of the enzyme glucose 6-phosphate dehydrogenase, from the glycolysis, the glucose 6-phosphate will be shunted to produce ribose 4-phosphate. Parallelly, NADP plus will produce NADPH, and the entry of the synthesis of ribose 5-phosphate marks the initiation of the pentose phosphate pathway. So this pentose phosphate pathway is produced as a shunt from the main glycolysis pathway, and it will generate from a hexose compound, so it is also known as hexose monophosphate compound, so it is known as hexose monophosphate shunt. So in a nutshell, we can say that the pentose phosphate pathway is not just about energy, it's about biosynthesis, protection and flexibility. It keeps the cell supplied with NADPH and essential sugars for life's vital processes. So my dear students, today we discovered how the pentose phosphate pathway or hexose monophosphate shunt plays a crucial role beyond energy production or by providing NADPH for biosynthesis reactions and ribose 5-phosphate for nucleic acid nucleotide synthesis. We learned how its oxidative and non-oxidative phases work together to maintain cellular redox balance, support fatty acid synthesis and protect cells from oxidative stress. Understanding this pathway helps us appropriate how intricately our cells manage energy, growth and protection. If you enjoyed this class, don't forget to like, share and subscribe for more engaging biology lessons. See you in the next class, and as always, stay curious and keep exploring the amazing chemistry of life. Thank you.